So if you look at the raw reads, again, without filtering the outlier cells, what we see is actually hard to say anything. It's a big cloud of points, right? So if you remember, I think with UMI counts, even with the raw reads, raw counts, the picture was better than this. So with the reads, because of amplification, the picture is always a bit messy, more messy than with UMI counts. If we plot now the log counts, so instead of using the raw counts, we use the log transformed counts. This looks already better. So we see there is some total feature effect on the first principal component. The variance is quite low here, and some batches are separated, and individuals separated from each other. Okay, now if we run Pca on log transformed counts, but on the reduced data set, so where we remove the outlier cells, this becomes even clearer. So again, we have this individual. This individual looks like always separated from others. These two individuals are together, and there is no total features effect. As you can see, the variance is just 6% on the first principal component, and we don't see this dependency of total features on the position on the first principal component. Now if we plot Pca plot on log counts of the unfiltered data, again we get some big cloud of points. This is the perplexity 130. If we filter out outlier cells, it still looks messy to me. So it doesn't tell you anything useful, I think. The same if we change perplexity to 10, change perplexity to 200 doesn't really change anything. So because the reads data is much noisier than UMI data, t-SNE fails to find any structure in your data. Whereas Pca was able to find some biological differences between the batches. So I hope you got some messages from these two chapters. There are some subjective steps in your analysis. So I think here we represent our subjective opinion about t-SNE, but it's not just objective. I mean, you can run it yourself. You can see that sometimes it doesn't really work as you want it to. But I think our main idea is just for you to know that and to treat t-SNE and Pca accordingly in the future. Any questions? All right. So the next chapter, Tallulah will be presenting the next chapter. This is more interesting and more biological now.
Not quite. Well, not quite biology yet. Tomorrow will be all biology, we promise. But first we have to make sure the data is good before we can do any biology in it. Otherwise, we're going to find the wrong biology. So here we're going to talk about identifying confounding factors that may be present in your data and how to quantify whether those factors are bad enough that you need to do some sort of correction to remove that confounding factor. So we're going to continue using the Blischak data, or the Tung data. So in the previous section, we looked at PCAs. So when we look at the PCA, you can notice that we have this strong correlation with the total features. So here we have the small points over here and the large points over here. The component 1 is largely driven by the total features, which we can see by looking at this plot, but it would be nice to be able to quantify that in some sort of way. So Skater has a function that allows us to correlate any sort of variable what or whatever we have associated with each cell with each of the different principal components to see what factor is driving that principal component. Yep.
No, that makes no difference to the functions.
So here's how we can apply this to look at whether the number of detected genes is correlated with the different principal components. So here we're actually using the total features. So that's the total number of detected genes. And here we're saying look at the BCAs. And we're going to use the log transformed counts. There. So here we can see that indeed component 1 is almost entirely driven by the number of detected genes, whereas the other components are much less so. Everyone understands that plot?
If you would like to define component 1 as a basic one, how would you define it? Component 1?
So component 1 is the component that explains the largest amount of variability in your data.
Yeah.
So here it's saying the largest source of variability is the total number of detected genes.
Yes. What I mean is there a way to define it in a way of the name of genes in
Oh, is there a way of
separations?
getting the loadings for the principal components out? No. No, not. So there isn't a way of extracting the loadings from Skater. Yes. Another way of looking at this is we can take each gene and do a general linear model and calculate the R² value for each variable we want to consider for each possible confounding factor, and then look at the distribution of those R² across all of the genes to see which factors are most contributing to the variability of each of the genes across the whole data set. So here we've got a list of variables. So some of these variables have been calculated by Skater when we did the quality control analysis. Some of them are annotations we've provided, so batch and individual are annotations. And then it will calculate the R² values. So here, this is the R² value, or the percent variance explained. This is the frequency across all of the genes. So here you can see the further to the right the peak is for each of these factors, the more total variance in our data set is being explained by that variable. So here we can see that the total features and the total counts are essentially the same and explain the most, followed by batch, and then individual. So individual is the variance we're actually interested in. We don't care about the variance that's just due to batch. So here we obviously need to do some sort of batch effect correction to reduce the variance that's explained by batch and preserve the variance explained by the individual. Any questions about this plot? No, good.
What is individual?
Individual, so these are cell lines from 3 different donors. So individual is the donor ID. So it's due to genetic background of the different individuals.
So how is that different than batch? Sorry, the batch.
Yeah, so for each individual there are 3 replicates. So each individual by replicate is a batch. So there's 9 total batches for 3 individuals. So there are other potential confounders, such as the cell cycle or apoptosis markers that you might want to correct for or you might not, depending on your particular biological system. For instance, if you're looking at cancer, obviously the cell cycle is a key part of the biological features of that system, so you want to keep it in that case. But if you're looking at some sort of in situ tissue, you probably don't care about the cell cycle that much. You're more interested in the other cell types, in which case you might want to remove the cell cycle. One method of removing the cell cycle or other features is SCLVM. However, this is currently not in R, so we can't show it to you today. But we've got the link to the paper if you want to check that out for yourself. So as an exercise, I'll have you repeat this analysis for the reads. I'll give you 10 minutes to work on that now. Okay, so I'm going to quickly go through the exercise, and then we'll have a 10-minute coffee break. So if you do the PCA for the reads, you can see that there's still a bit of a trend related to total features in the principal component plot. And then when you look at this plot looking at the correlation, you can see that there's still a strong correlation between the principal component 1 and the total features, but it's less strong than with the UMIs because there's additional noise due to amplification. And then if you look at the confound this plot, you can see that batch is more important relative to total features, or total features in total counts is less important than it was for the UMI data. But otherwise, we still have a strong batch effect and still have a strong effect for total features. And that's it. So now we'll have a 10-minute coffee break, and we'll continue again.